Hello dear students, welcome to yet another session of AIATS Plus Phase 2, where we discuss the questions in which most of you tend to make mistakes. And this discussion is particularly of the mock test 8, which was held on 9th of March. I am Snehil, your zoology faculty at Aakash, and as you are aware that there are certain topics from which we are going to discuss the questions, as this has been our ritual ever since the beginning. So the topics that I am going to take up are biomolecules, excretory products and their elimination, locomotion and movement, and these 3 chapters pertain to class 11th. And then we have human health and disease and evolution, and these 2 chapters pertain to class 12th of our syllabus. And as I always mention in my session, that biomolecule is one such chapter from which many questions are being asked in the NEET exam. That is what our analysis tells us. So let us begin the discussion without wasting much time. So the first question that I see on my screen today is question 140. And it states, read the following statements and select the correct option. So basically we need to read the statements and then make out whether these statements are correct or not. So the first statement states, in the structure of a glycogen molecule, a free aldehyde group is present at the left end of polysaccharide chain. And the statement 2 states, starch and cellulose can be distinguished on the basis of iodine test, as cellulose gives blue color and starch gives red color with iodine. So for solving this question, we need to recall the concepts of carbohydrates, as I can see the names of different carbohydrates. And particularly we need to focus on their structure to deal with such questions where we are talking about whether there is a aldehydic group or not, and we are talking about whether there is a particular chemical test in which certain color is formed. So let us quickly recall the concept of carbohydrates. So we all know that carbohydrates can be of different types, and they are monosaccharides, oligosaccharides and polysaccharides based on the number of monomeric units they contain. All right. So basically we need to focus whether there is a presence of any free functional group or not. So where does that comes into picture? So basically monosaccharides, for instance glucose, if I draw the structure of glucose, we will see that we have a free group which can participate in further reactions. Let me draw the structure of glucose for you. So here you can see the structure of glucose. All right. In this structure, on the 3rd carbon molecule, you can see that we have a free carbonic group which can act as a reducing agent. So basically the presence of free carbonic group like aldehyde and ketone contribute to the fact that this particular molecule can act as a reducing agent, and it will reduce another component which is present in a solution. For instance, when we are carrying out different chemical tests, for example, we have the Benedict's test, Felling's test and etc., Tollens reagent test. So basically what happens, for instance, if we carry out Benedict's test by keeping glucose, the group present at the third carbon atom, the free group present, will reduce the copper ions present in the Benedict solution into cupric form, and it will turn the red color of the Benedict solution into blue color. It will convert the blue color of the Benedict solution into red color due to the presence of a reducing group. So this is how we identify that whether there is a presence of a free group or not. If there is a reaction in the chemical solution, it will indicate that there is a free group which is interacting, and we can call it as a reducing sugar. So we say that all monosaccharides are reducing in nature. So we say that all monosaccharides like glucose, fructose, ribose, etc. are reducing in nature because they have a free aldehyde or a ketone group. But in the question we were asked that whether glycogen has a free aldehyde group or not. It was stating that there is a presence of a free aldehyde group. But if you look at the structure of glycogen, which is a polysaccharide, we can see that there is no free group which can interact or which can act as the reducing agent. So basically in polysaccharides, the left end represents the reducing end, the left end represents the non-reducing end, whereas the right end represents the reducing end. So whether a polysaccharide will be reducing or non-reducing in nature, that is a debatable case, whereas all monosaccharides are reducing in nature. So we can see the structure of glycogen, and if we go back to our statement, it states that a free aldehyde group is present at the left end, which will be incorrect because free aldehyde group will be present at the right end, which represents the reducing end. So here it should be the right end and not the left end. So this helps us conclude that our first statement is incorrect. So now we go back to our discussion again to figure out whether the second statement is correct or not. So let us quickly see the structures of different molecules such as starch, which is given in our question. Then we have cellulose mentioned in our question as well, and we are already discussing about the glycogen. So basically starch is made up of two components, we can see, amylose and amylopectin, and starch is said to have secondary helices in its structure which helps it trap the iodine molecule when we are trying to perform a test. Alright, so there are secondary helices which trap the iodine molecule when we are performing the iodine test, and this gives a blue color complex. So we say that the starch iodine complex forms a blue color because the iodine molecules get trapped in the secondary helices of the starch molecule. And it is said that when we try to react cellulose with iodine, there is no such color formation. So why is it happening? Why do we have such observation when it comes to cellulose? That can be explained by the fact that we can see that cellulose do not have a secondary helical structure. So we can say that they lack secondary helices in their structure, and therefore we can see it has more of a linear structure, and therefore since there is no helical like structure which can trap the iodine molecules in between, and thus we can say, and thus we see no color formation because the iodine molecules are not getting trapped. So basically when we try to perform a reaction between cellulose and iodine, there will be no such color formation. Whereas if we talk about glycogen, we can also see that glycogen has a branched structure, and the branching point in glycogen is formed by the alpha-1,6 glycosidic linkage, and we can see that it has a branched structure. So it is said that if we try to interact with iodine, then there is a formation of a red color complex. So basically the formation of color complex is depending on the fact that whether secondary helices are present in the structure of the carbohydrate or not. So we can see very clearly that secondary helices are present in the structure of starch and glycogen, and therefore they are giving that colored complex, whereas the structure of cellulose is not branched, and therefore they cannot trap the iodine molecule. So now we go back to our question to figure out whether it is correct or not, and we can see, let's read it again. So we can see that it is written that starch and cellulose can be distinguished on the basis of iodine test, which is absolutely correct, that we can distinguish them, as cellulose gives blue color. So this helps us realize that this statement will stand incorrect, because the later part of the statement which states that cellulose gives blue color is incorrect, as we already discussed that there will be no color formation, and starch gives red color with iodine. So we discussed that it is a glycogen which gives red color complex with iodine, not starch. Rather, starch iodine complex will form blue color. So overall we can make out that the second statement stands incorrect. So our final answer will be that both statement 1 and 2 are incorrect, and we will straight away eliminate the other options. So this was a simple question only based on the fact that we need to analyze the structures of different carbohydrates, and it is straight away from the NCERT itself, although it looked complicated, but it was not. So we move on to our next question, and I can see that students have joined us in the comment section. A very warm good afternoon to everyone. I hope you are enjoying the session, and please ask questions if you have any doubts. So we move on to our next question, and the question here is question number 141. Again we can see a structure, and it pertains to the chapter of biomolecules. So here comes the fact that biomolecule is a very important chapter from which many questions are asked, as I have been repeating myself in every session. So we can see the structure of a molecule, and then what we need to do is we need to choose the incorrect statement with respect to the compound shown above. So our first task here is to identify the given molecule. So here I can recall from the chapter of biomolecule that this structure represents the molecule of cholesterol. So this also helps me figure out this fact that each and every diagram or table given in the chapter holds equal weightage, because we can be asked questions based on it. Although the question might be tricky, it can be easy also, but if we are not aware with the diagrams, we will straight away get afraid when we see the question and will have a tendency to leave the question. So it is always a good way to solve questions based on NCERT figures, so you don't make the mistake of skipping those by considering that they are not important. So we can see that this is the structure of cholesterol, as I already said, and this diagram is very much taken from the NCERT. So now we need to discuss few important points when we talk about cholesterol. So basically, lipids, cholesterol can be categorized as a type of lipid. So we know that there are different types of lipid, and they are categorized as simple lipids. Then we have the conjugated lipids. And then we have the derived lipids. So simple lipids are basically lipids which contain glycerol and fatty acids, and the famous example for this, which is also given in the NCERT, is the arachidonic acid and the palmitic acid. And we say that arachidonic acid and palmitic acid are true lipids. All right. Arachidonic acid has 20 carbon atoms, we know that, and palmitic acid has 16 carbon atoms, including the carboxyl atoms, as the NCERT states. So we are often asked question based on these numberings as well. Now coming to the conjugated lipids, the example which is given in the NCERT, although they have not mentioned the word conjugated, but it is our duty to read in between the lines when it comes to NCERT, because it tries to explain a lot of things within a single sentence. So basically, conjugated lipids are the lipids in which other groups like metal ions are attached. For instance, the example that we have here is lecithin. In lecithin, we have the fatty acid in which phosphorus is present. And the example of lecithin is given in the NCERT, and you can be asked that whether it is a conjugated lipid or not in the form of question. So it is a conjugated lipid, all right. Then we come to our derived lipids. So here comes the example of cholesterol. So basically, cholesterol is a derived lipid. Basically, the derived lipids are formed from steroids, and under steroids we have a category which is known as the sterols, and one of the famous sterols is the cholesterol. All right. And we can see that it is made up of 4 fused rings. So question can often be asked in this way, that how many rings are present in the structure of cholesterol. So we can say that there are 4 rings present, and it is important to note here that they are 4 homocyclic rings. This is also one of the words which is used in the question to complicate the question, that whether these rings are homocyclic in nature or heterocyclic in nature. So basically, when in the ring, if there is a presence of a carbon atom, if there is a presence of any another atom except for the carbon atom, for instance, if in a structure we have carbon, along with it we have a nitrogen, we say that it is a heterocyclic ring, because that is the presence of another atom along with the carbon atom. For instance, the examples of these are the nitrogen bases, whereas here it is the homocyclic ring. As you can see that all along we have the carbon atoms. And now coming to a few properties of cholesterol. So it is one of the most abundant steroids in the human body, and it is formed in the liver. And further, let's go back to our question to figure out our answer in terms of cholesterol. So the first option states that it is the most abundant steroids in animal tissue, which is absolutely right. Cholesterol is also present in the plasma membrane. Secondly, it states it can be synthesized in liver, which is also true, as cholesterol is synthesized in the liver. Therefore, option 2 cannot be the right answer, as it is a correct option. Moving to option 3rd, it is stating that it is water soluble organic compound. Now, one of the important properties of lipids is that it is not water soluble. In fact, in the NEET previous year's papers of 2021, it has been asked that whether lipid can be dissolved in an organic solvent or not, and the answer is yes. And we say that lipids are not soluble, not water soluble, but they can be dissolved in organic solvents. And the examples of organic solvents can be chloroform. So basically, like dissolves like, it works on that concept that in organic solvents like chloroform, lipids can be dissolved. So by stating here that it is a water soluble organic compound, it will be an incorrect option, as it is not water soluble. So option 3rd is probably the answer that we are looking for here. But let us read the last option as well before we conclude anything. So the last option states its deposition in arteries can cause atherosclerosis. So dear students, we know that atherosclerosis is a disease which is associated with our heart. So basically what happens is that we have our arteries, and this is the wall of our artery in the normal condition. But when there is increased level of cholesterol in our body, then what happens is that these arteries undergo thickening due to the deposition of the cholesterol. So basically this cholesterol gets deposited in these arteries and they thicken and form a plaque. And obviously since the wall of the arteries have thickened, we can see that the passage of blood flow here will be reduced, or we can say the lumen becomes narrow and it will obstruct the flow of blood. So this condition gives rise to a heart condition which is known as the atherosclerosis, and therefore we can conclude that our fourth option is also absolutely correct and cannot be the answer, as we are looking for the incorrect statement, and therefore our right answer here is option 3rd. So you can see that from a simple diagram which is given in the NCERT, how the concepts can be interlinked, and we can find this question. I'll tag this question as difficult because there are 2 to 3 concepts which are aligned to this question. So this helps me figure out that we need to read the NCERT very carefully, keeping in mind that we need to focus on the diagrams as well as the in-between lines. Also, we need to learn how to apply different concepts together, because we will study about atherosclerosis when we study the chapter of body fluids, but it has been linked to the chapter of biomolecule in a nice way. All right. So now we move on to our next question, and it is question number 145. And the question here states, which of the following is, someone is asking in the comment section, Ankul Kumar, why 4 is wrong? So Ankul, you are right that the fourth option is correct, but in the question we are looking for the incorrect option. So we'll go with 3 because fourth is correct as you are stating yourself. Okay, so we move on to our next question, which is question 145, and it is stating which of the following is incorrect for an exothermic reaction in presence of biological catalyst. Alright, so we discussed the concept of exothermic and endothermic reaction in our previous class as well, but let us quickly recall the concept. And the figure here that you see on your screen is taken from the NCERT. So this is again a good example of how a figure based question can be asked and we can find it as a difficult question. Alright, so here we can see a graph which is showing us the potential energy level and the progress of reaction. And over the course of reaction, we can see that we have our substrate which is being converted into the product. So the substrate here, when we talk in terms of potential energy, is at a higher value as compared to the product. So here, although it is not mentioned in the NCERT, but by looking at the levels of the substrate and the product with respect to the potential energy, I can make out that this diagram is illustrating an exothermic reaction. So how I was able to conclude this? Let us try to understand it by recalling the theories which we discussed in our previous classes as well. So basically reactions can be categorized into exothermic and endothermic based on whether the reaction requires heat or it releases heat. So basically we have two types of reactions. They are the endothermic reaction and the exothermic reaction. And in the endothermic reaction, heat is given to the reaction. So basically heat is supplied in this case, whereas in an exothermic reaction, the heat is given out. Heat is released, we would better say that heat is released. That is why we can see that the substrate is at a higher potential energy, but when the reaction will occur, there will be release of heat, and then we'll see that our product has a lower value of energy. Alright. So here in endothermic reaction, the substrate will be at lower level with comparison to the product in terms of potential energy. So had it been the diagram of an exothermic reaction, then we can say that here will be, had it been the diagram of a endothermic reaction, then my substrate would have been at a lower level, whereas the product, let us draw it separately for better understanding. So if the diagram was for an endothermic reaction, then if we mark potential energy here, our substrate will be at a lower level in comparison to the product. So you can see the difference between the two diagrams here. In the endothermic reaction, the substrate is at a lower potential energy in comparison to the product, whereas in the exothermic reaction, it is at a, the substrate is at a higher energy level. So this is the basic difference between the exothermic and the endothermic reaction. Okay. Also, so we see our question now, and we can see that in the question there are also references of the mechanism of enzyme action. So let us quickly recall the mechanism of action of enzymes. and we can see a figure here. So the first thing that happens during an enzyme reaction is that our substrate will try to bind to the active site of the enzyme. As we can see in the figure, we have our enzyme here and our substrate will bind to the active site. So when the substrate binds to the active site, then there are conformational changes, there are rearrangement and breaking and formation of new bonds. All these leads to the binding of the substrate to the active site, and there is a formation of enzyme substrate complex. And the formation of enzyme substrate complex, although a transient phenomena, we say that it is a transient phenomena, which means that it is a temporary phenomena, but it is very important. It is one of the very important steps or a mandatory steps, which means that there will be no reaction where there is no formation of enzyme substrate complex. All right. So this formation of this complex is a mandatory steps. So further, when enzyme substrate complex bind to each other, then there will be interaction between them, there will be another conformational changes, and here finally we have an enzyme product complex now, which is again a transient step, and from the enzyme product complex there will be further breakage of bonds and the products and the enzyme gets separated. And then finally we have our product, which was our desired product, and then we have our enzyme, which is free again to be used. So we say that in the end of the reaction the enzyme becomes free and we can use it again, or the enzyme can be reused after the reaction. So I can see in the comment section, Srinath is asking that enzyme substrate complex is formed before formation of enzyme product complex. So Srinath, you are absolutely right. The enzyme substrate complex will be formed first and then the enzyme product complex is formed. All right. So we now go back to our question to figure out our answer. So again we need to find the incorrect option with respect to the exothermic reaction. So my first option states enzyme decreases activation energy for a reaction. This is absolutely true and cannot be the right answer. Whether it is an endothermic or an exothermic reaction, we know that the enzymes are known as the biocatalyst, which means they increase the velocity of the reaction. And how do they do it? They accelerate the rate of reaction by lowering the activation energy which is required for any reaction. For any reaction to occur, activation energy is required, which is the energy between the substrate and the transition state. And we can see that we have the activation energy at a higher level without the enzyme, whereas in the presence of enzyme the level of activation energy has decreased. So whether it is an exothermic reaction or an endothermic reaction, the enzymes are going to lower the activation energy. Therefore first option cannot be the right answer. Moving to the second option now, potential energy of the product is less than that of substrate. So as we already discussed at length that the product will be at a lower potential energy when we talk about the exothermic reactions. Therefore option 2 cannot be the right answer. Moving to the third option now, which states enzyme increases velocity of reaction. So as we already discussed that enzymes are biocatalyst and it is their role to increase the velocity of reaction by decreasing the activation energy level. Therefore option third also cannot be the right answer. Then lastly we have the fourth option, which states enzyme substrate complex does not pass through transition state and product is formed immediately. So dear students, this is an incorrect option, as we already discussed that there is a formation of enzyme substrate complex. So dear students, for progression of any reaction there will be various formation of altered structure, which are known as the transition state or the transient phenomena, because they are temporary structures, but it is mandatory that during the formation of the product from the substrate, there will be formation of the transition state. There will be no reaction where there will be no formation of enzyme substrate complex, or where there will be no formation of transition state. So, the given option is incorrect and the answer that we are looking for in this question. So, I hope you are able to solve such questions now based on the figures when it comes to our NEET exam, because in the NCERT, although they have just given the figure, they have not talked about it in great details, but we can see that questions can be asked based on it. Right. So, we move on to our next question now, which is question 171, and it states the component of cell that constitutes more percentage of the total cellular mass than proteins is. So, dear students, it is a very simple question, but one can go incorrect in this only because of the fact because we failed to focus on the table which is given in the NCERT. So, here you see a table which is very much taken from the NCERT, and we can see that we have the different components given which constitute our cell, and then there is the percentage of the total cellular mass. So, this question can be solved in a jiffy only by remembering these certain values. And here the question that they are asking is the constituent of which component is more than proteins, and we can very clearly see that it is water. All right. So, in the options given, we can see a third option states water, and therefore, the right answer will be option third. So, although it is a simple question, but students can make mistakes only based on the fact that they did not focus on the tables which is given in the NCERT. All right. Now, we move on to our next question, and it is again from the chapter biomolecules. And the question states, the retentate obtained after straining the slurry made by grinding a living tissue in trichloroacetic acid contains which set of compounds. So, dear students, from the question we can make out that here we are talking about the concept of acid soluble and acid insoluble pool. So, let us quickly revise the concept of analysis of the living tissue. So, basically when we perform the analysis of a living tissue to figure out the chemical composition of that tissue, there is a particular step that we follow, and it is already mentioned in the question that a slurry is made of the tissue, and then in the trichloroacetic acid, question can also be asked in the exam that which chemical is used to make the slurry. So, it is trichloroacetic acid in which the slurry is made, and then it is made to sieve through a cheesecloth. So, it is just like the process in your kitchen where you make tea and then you try to strain it on a strainer. So, just like that, when we are straining the slurry which we get two fractions which are named as the acid soluble fraction and the acid insoluble fraction. So, basically just like if you recall the example of making a tea in your kitchen, you can figure out that you can remember that when we make the tea and we are trying to strain it through a strainer, the fluid part will go down into the cup, but the tea leaves or the ginger that we added stays over the strainer. This is exactly what is happening here. So, basically the acid soluble pool is the filtrate part, or you can remember it that it is the tea part when we are making the tea, whereas the acid insoluble pool is the retentate part or the part which will stay over the strainer. And how it is happening here? Just like it happens the way when we are making the tea, that the larger molecules will stay over the strainer, whereas the molecules which is smaller in size will go down the cup. So here the molecules which are obtained in the acid soluble pool are basically the molecules whose molecular weight ranges about 800 to 800 Daltons. It is the approximate value. Whereas the molecules which are obtained in the acid insoluble fraction, they have molecular weight which ranges in more than the 1000 Dalton. So it is very obvious that the macromolecules or the molecules which have larger size will be present in the acid insoluble fraction. So here the molecules that we are getting are the macromolecules. They can also be called as the macromolecules. Whereas in the filtrate, the molecules that we get are the micromolecules. So in the macromolecules, we have the polysaccharides, we have the proteins, we have the nucleic acids. But one exception here again, as biology is full of exception, there is the exception of lipids. We say that lipids are not macromolecules very precisely, as their weight is generally lower than 800 Dalton, but still they are found in the acid insoluble fraction. And why does that happen? It happens because when we are trying to break down the cell membrane, and we already discussed that our cell membrane is composed of lipids when we were discussing the question on cholesterol. So when we are trying to break down the cell membrane breaks down and forms small vesicles which are water insoluble vesicles. So basically these water insoluble lipids are present in the form of water insoluble fractions, as they form water insoluble vesicles, not fraction, I'm sorry. They are present as water insoluble vesicles, and thus they separate out in the acid insoluble pool. Whereas in the acid soluble pool, we will have the molecules like the phosphate, the ions, or the monosaccharides, the amino acids, the simple amino acids. Those things will be found in the acid soluble pool, as they have lower molecular weight. So just keep the example of tea in your mind, that they will go down the strainer very easily inside the cup. So now we go back to our question. So my question here is about retentate. So we are talking about the acid soluble pool. Sorry, we are talking about the acid insoluble pool. So let us read the first option. So my first option states proteins, minerals and lipids. So here, since we can see that we have minerals, so minerals will be found in the filtrate or the acid soluble fraction. Therefore option 1 cannot be the right answer. Moving to option 2, which states phosphate ions, sulfate ions and magnesium ions. So these will be found in the filtrate fraction, not the retentate fraction, and therefore option 2 also gets eliminated. Then we have the third option, which states phosphate ions and sulfate ions, then the proteins. Then this is again an ambiguous option, because here proteins will be found in the acid insoluble fraction, but the other two will be found in the acid soluble fraction. So this option also stands incorrect. Then lastly we have the fourth option, which is probably answer. As we can see, we have proteins, nucleic acids and the polysaccharides, which will be found in the acid insoluble pool or the filtrate or the retentate, I'm sorry. And thus our final answer is option 4. So now we move on to our next question, and here we can see our next question is question 161. And the question is stating, select an opiate drug which suppresses brain function, relieves intense pain, and thus is used as an analgesic post-surgery. So there are very heavy terms in this question. So we can see that we are talking about an opiate drug which is suppressing the brain function and it is an analgesic. So there are 3 keywords which will help me find my answer, although the question pertains to the topic drugs that we study in our chapter Human Health and Disease. And dear students, it is important that you know that that portion of the chapter is very important when we talk in terms of NEET exams, because several questions are asked based on the chemical nature of the drugs, their effects and their figures. As there we can see the figures which is taken from the NCERT itself. We have various figures in that portion and questions are very frequently asked from those figures. Alright. Okay, so the figure that you see here is the structure of the opiate drug that we were talking about in the question. So let us first discuss about drugs. So drugs can be of various types and they are based on their chemical nature. We have the opioids. Then we have the cannabinoids. Then we have the alkaloids. And based on their effect, drugs can also be categorized as the stimulants. Stimulants are often given. Stimulants are basically stimulate our nervous system. So basically stimulants are given in the case if a person is suffering from depression. So stimulant, there can be hallucinogens. Hallucinogens are basically drugs which alter our perception, the way we think, and they give us a sense of good feeling. So basically we are hallucinating, we are seeing things which do not exist. So we have stimulants, we have hallucinogens, we have sedatives, then we have the depressants. So basically we have various categories and drugs are categorized in these different categories based on their effect or action. So here let us go back to our figure and we saw that here it is a picture of a plant and a chemical structure. So basically the chemical structure here represents the molecule of morphine. Here the structure is morphine. And the plant that we are seeing in the figure is the poppy plant. So morphine is basically a type of opioid. So we are going to quickly revise the opioids. So basically opioids act via the opiate receptors which are present in our brain. So questions are often asked based on whether these receptors are present in our body. So they are present in the brain as well as in the GIT tract. So they interact via the opiate receptors, and the famous examples that is given in the NCERT for opiates are the morphine and heroin. So we say that morphine is an analgesic, which means that it helps alleviate pain. It is a pain relief. It gives relief from pain. So morphine is often used by doctors after surgery so that the person doesn't experience extreme pain. So morphine is an analgesic, whereas we say that heroin is a depressant. So this is an important difference between the two, that heroin is a depressant whereas morphine is an analgesic. So morphine is obtained from the latex of poppy plant. So it is important that we know that morphine is obtained from the latex of the poppy plant, which is also known as the Papaver somniferum, and heroin is formed from the diacetylation of morphine itself. So diacetylation of morphine will form heroin. Heroin is basically a bitter crystalline substance and it is abused as an depressant by various drug abusers. So this is the basic about the opioids, and it is often taken by injection and inhalation. This is also an important point on which question can be asked. Moving on to our next category that we have, the molecule that you see here is the cannabinoid molecule. And the plant that you see here is a leaf, and it is a leaf of the cannabis plant. So basically, cannabinoids are naturally occurring substances in the cannabis plant. So we are talking about the cannabinoids now. So cannabinoids are basically obtained from the inflorescence of the cannabis plant, which is known as the cannabis sativa, and we say that the leaf, flower top, the inflorescence involves most of the parts of the plant, and they can be used in various combinations to produce drugs like charas, ganja, marijuana, etc. And all these drugs affect our brain as well as the cardiovascular system. So it is important that we know that these drugs are affecting our brain as well as the cardiovascular system. And these drugs can produce hallucinogen at higher doses, and they do not hold any significant clinical application. But these cannabinoids are also often used by various sports person, and in general in general it is illegal to abuse these drugs. All right. So we move back to our question now. So in our question we can see that there is another name, which is the LSD. So LSD is basically a drug which is obtained from the fungus known as from the fruiting body of the fungus known as the Claviceps purpurea, and it is a very potent hallucinogen. So basically, when we are studying the topic of drugs, the key points that we need to remember is its source, where are the receptors present for its action, and what are the plant part which can be used for its production. For instance, the morphine was being produced from the latex of the poppy plant, whereas the cannabinoids are obtained from the inflorescence of the cannabis sativa. And LSD is produced from the fruiting body of a fungus, which is the Claviceps purpurea. All right. So in the question, since we have already discussed that we are looking for an opiate which will suppress brain, as morphine can act as a depressant, and it is an analgesic. Analgesic means it is a pain reliever. So the answer here that we are looking for is the morphine, and we will eliminate the other options. So dear students, this marks the end of the discussion of the zoology portion when we are talking about the mock test 8 exam. But hold on, as we still have to discuss the portion of the botany for which our esteemed faculties will be joining you soon. So stay tuned and all the best. Hope to see you again. Good luck for the upcoming NEET exam. Thank you.